So what I'll be focusing on specifically is reliability of clinical trials. And as everybody knows who has worshipped at the shrine of the ICH E6R2 guidance document, as well as the preceding FDA risk-based monitoring guidance document, the idea that we should be doing risk-based monitoring and related activities for the purpose of improving the reliability of clinical trial results is mentioned throughout those documents. It's actually mentioned 11 times in the ICH E6R2 document, and it's also mentioned in the academic literature on risk-based monitoring, which is of course quite limited, but it's still there. So what I will discuss today is, if you read the ICH E6R2 document, though, you will not find a definition of the word reliability. You will not find a definition of the word accuracy. You will not find any hint whatsoever about how to know whether you are, in fact, improving the reliability of your study results. It's just kind of in the water. It's assumed that everybody knows what that means and that somehow we'll know it if we achieve it. It cannot be assumed, and you cannot know if you are achieving it unless you have a specific way of thinking about that. And that's what I will talk about. That's what I will offer you today, and I'll be very interested in people's feedback and if others have different ways of looking at it. I do think that it might interest you to know how I got involved with this in the first place, because I was a clinical neurologist and pain management specialist working in a cancer hospital and working in a non-cancer hospital. And I was really only interested in taking care of patients suffering with the diseases, and really at the beginning couldn't care less about clinical trials, and really wasn't even that interested in developing new therapeutics until time went on, and I realized that the tools that we have to treat patients are really pretty primitive and haven't really changed much over thousands of years, and are causing a lot of toxicities. And that's what propelled me to get interested in clinical research. And even then, I was working as an investigator at a clinical research site that I set up here in Boston. And even then, I wasn't really that interested in clinical research methodology until I started to have pharmaceutical companies knock on my door and walk into my office and say, "Hey, Dr. Katz, why did my clinical trial fail?" And my initial reaction was, "What do you even mean that your clinical trial failed? What are you talking about? How can a clinical trial fail? If your drug works, then the trial would show it." And of course, and I'm sure that all of you on this call understand how naive that was, but I didn't at the time. And so these companies would come to me and lay this corpse of their clinical trial down on my table and ask me to dissect it and figure out what the cause of mortality was for their clinical trial. Morphine versus placebo, P greater than 0.05. We know that can't be right. Ibuprofen versus placebo in dental pain, P greater than 0.05. That can't be right. So clearly it was the trial that had failed and not the drug in those cases. And I became a pathologist of clinical trials, trying to dissect the sources of failure, which I had no idea at the beginning, what might actually cause a clinical trial to fail, because of my naive idea that I think truly most clinicians have, and I think actually many pharmaceutical companies have, that if your drug works, then your trial ought to show it. Probably not the people on this call, but I assure you that that idea is still prevalent out there. So this sent me on a 20-year quest to try to answer this question: why do trials fail? I don't have to tell you on this phone, especially those who work at pharmaceutical companies, that this is a disaster. If you have enough money in the bank, then you might have to repeat your study again. It's a massive waste of resources. Not everybody has enough money to repeat their trials 2 and 3 times, and therefore programs may die prematurely that ultimately might have actually brought a new therapeutic to patients suffering with diseases that need them. That's bad. Worst of all, I think, though, is that if we're experimenting on human subjects and we are doing it in such a way that our trial is destined not to generate scientifically useful data, or to put it a different way, not to generate accurate and reliable results, that's inherently unethical and violates every ethical guideline. So we don't want to be doing our trials in a way that they're destined to fail, which therefore compels us to understand why they do fail. Another way of talking about failed clinical trials is to talk about reliability of clinical trials, because, to put it another way, clinical trials fail when the methodology is unreliable. And that's why regulators are so concerned about this, because they know, although they didn't reveal much about their knowledge in these guidance documents, that it is unreliable research methodology that causes clinical trials to fail, hence their emphasis on using our methodologies to improve the reliability of clinical trials. Well, if that's the case, it might be a good idea to have some sense of what reliability actually means and how reliability can become problematic and cause clinical trials to fail. So even though the definition of reliability is not anywhere in these guidance documents that we're following, it's everywhere else. And so, for example, there's a organization called the Joint Commission on Metrology that is an umbrella organization of multiple other scientific organizations that got together a few years ago just to achieve consensus terminology on words related to measurement. And if you haven't seen that paper, I implore you to review it so that you can make your own use of terminology more reliable and more accurate. So reliability has a definition. It means how close repeated measurements are to each other, and accuracy is its first cousin, or maybe its fraternal twin, which means how close a measurement is to its true value. So if you're doing dental pain studies and you're measuring the effects of ibuprofen 400 milligrams for the treatment of dental pain, and you do 10 clinical trials, and each time you get virtually the same result for the efficacy of ibuprofen versus placebo, you're doing reliable work. The way you do clinical trials is reliable. Whereas if you get numbers for the same effect of the same drug for the same kinds of patients for the same disease at the same dose measured the same way, and your results are different from each other, then you were doing an unreliable job measuring the efficacy of that drug using that research methodology. Similarly, accuracy means how close the measurement is to its true value. So if we can agree on what the true value is for the efficacy of ibuprofen in dental pain, which might be a standardized effect size of 1.2, which is actually about what it is. If you do a clinical trial and you get 1.2, well, that's accurate. If you get 3.2, that's not accurate, or 0.2. So these notions of accuracy and reliability, which appear ubiquitously in engineering and biology and other areas of sciences, including pharmaceutical manufacturing, by the way, we can apply these same concepts to our clinical trials and therefore get to the heart of what regulators are expecting from us. The typical way that you see these concepts of accuracy and reliability portrayed in the scientific literature is with this arrow diagram. So if you imagine each one of these dots on one of these targets as the result of a single clinical trial, and you imagine the center of the bullseye as being, let's just call it the true value, which of course usually we don't know, but let's put that aside for a minute. Then this is what we want in the upper left. We want each clinical trial to produce a result that's close to other similar clinical trials, hence reliable, and also near the bullseye, hence accurate. And I'm sure that you all understand what the other targets represent, which I won't go through. Well, if we maybe I'm being too hopeful, but if we do share a common understanding of what the word reliability, mentioned 11 times in ICHE 6R2, means, then we can actually start asking the question of whether clinical trials are reliable or not. If they're reliable, then we can ignore these aspects of the guidance. The regulators are wasting our time and money. If clinical trials are in fact not reliable, well, then we can think about quantifying their reliability, because if we can do that, then that might help us measure whether our efforts to improve reliability are successful or not. Because if in fact we are going to follow the advice of the regulators and try to improve reliability of clinical trials using these methods, and we're going to spend a whole lot of money and a whole lot of time doing it, maybe it would be a good idea to have some way of knowing whether we're successful. So are clinical trials reliable? Well, here they're not. And you can discern that looking at any meta-analysis of any treatment for any condition. If you're doing a meta-analysis, then by definition, you're making a decision that the trials in your meta-analysis are similar enough that they should be producing a similar result on the same drug for the same condition. The protocols are similar, the outcome measures are similar, the diseases are similar, the patients are similar, the regions are similar, et cetera. This happens to be acetaminophen for the treatment of osteoarthritis of the knee. It doesn't matter. You see the same thing when you look at any meta-analysis. And if you use standard metrics for the measurement of reliability of any measurement instrument, and I'm considering each one of these trials a measurement instrument, you can look at coefficient of variation, you could look at range, you can look at standard deviation, you can look at it in all these standard ways. You can see that no matter how you measure it, the reliability of clinical trials is poor. And you might even use the term inexcusably poor. And I can tell you that there's been very little attention paid, although there has been some attention paid, and I'll show you some data. There's been very little attention paid to why the results of our clinical trials, which should converge on some true value, careen all over the place, from numerically positive and statistically significant to numerically positive and not statistically significant to actually placebo looking better than active drug, which is common and which is not uncommon in meta-analyses. So clinical trials are not reliable. If you want to look at a different paradigm for evaluating reliability of clinical trials, you can look at so-called replicate clinical trials. Some of you are old enough to remember the days of replicate clinical trials, as I do, which are still occasionally done, where you literally take the same protocol and you just put a different protocol number on it, and you hand it to two different groups of investigators led by a different principal investigator, and you literally execute the same exact study in two different groups of sites, and you try to get the same results. And then if you do, then you can achieve the regulatory mantra of 2 adequate and well-controlled trials and get your drug approved. Well, in reality, replicate trials usually actually give you quite different results. And this is just an example from the pain literature, which is where I spend most of my time, showing that even the placebo group in red varies wildly from one trial to another, let alone the responses to active drugs. And in this particular case, one trial was positive, one trial failed, and the program was killed. This is lamotrigine for painful diabetic neuropathy, and this is not unusual. You can't look at this and say that the way that we do clinical trials is a reliable way of measuring the efficacy of drugs. And I support the regulators in urging us to develop methods to improve their reliability. And the only question that remains is, how do we do that? And how do we know whether we're actually accomplishing it? Well, if you're going to set forth on this path, as I have, then you need to ask yourself, why are clinical trials unreliable? It's the same reason we do root cause analyses in any other industry. If you can't figure out why Navy pilots crash their planes into the ocean, you can't do anything about that, which is actually an area of very active investigation. If you can't figure out why chemical plants explode, then you can't do anything about it to prevent them from exploding in the future. If you can't figure out why clinical trials are unreliable, you can't make them reliable. So 20 years ago, when I first got interested in this, there was virtually no literature on this topic, and everybody was flying blind. Now, there actually is a literature on this topic, and here's some representative publications down on the bottom of the slide. There are plenty more, and here are some of the reasons on the left side of the slide why clinical trials are unreliable. And if you're not aware of these things, you're not in a position to do anything about the reliability of clinical trials. First of all is reliability of measurement of clinical outcome assessment. It turns out that if you think that because a symptom intensity report, whether it's a pain score or a depression score or a sleep score or whatever somebody is reporting, that might be the primary endpoint of your clinical trial. If you think that everybody's subjective reports are inherently reliable because it's subjective, or alternatively, if you think that you can't evaluate whether somebody's symptom report is reliable or not because it's subjective, then you would be wrong. And you would be ignoring a whole body of literature that's come out now that shows that, in fact, the opposite is true. You can actually know whether somebody's subjective report is reliable or not, and you can actually correct it without having any idea what their true symptom intensity is. So if you're interested in reliability of clinical trials, then that is a literature that you need to familiarize yourself with. And this is just one example of that on the right, where we know, for example, that patients whose pain scores over time are close to each other compared to pain scores over time that are wildly different from each other. Most of this high variability in the example that you see on the right is actually error variance, and we know why that is. And we know that we can train patients to reduce their error variance, which actually reduces this variability. And we know that once we do that, we can now see differences between drug and placebo that were invisible before that kind of training. So this is all understood now in the literature. And this can be, and for you out there doing risk-based monitoring, you can be monitoring this. You can be setting thresholds and you can be raising alarms when this occurs. And I encourage all of you to start figuring out how to do that if you're not doing that already. The placebo response is another source of unreliability of clinical trials. The placebo response varies dramatically from one trial to the next. We have pretty good ideas these days about what contributes to what drives the placebo response, which is patient expectation of benefit, actually. And there are other things too, but that's the main one. And we know from randomized controlled trials that if we move from an environment where patients are allowed to have uncontrolled levels of high expectation, which you see here on the very right of this screen, and we do training and take other methods to control that expectation such that patients have more accurate expectations of what they're getting into, we can suddenly see emergences of differences between drug and placebo of drugs that we know are efficacious that were lost when we let that be subjected to the chaos of the normal clinical environment. So those are just two examples. I don't have, this is a whole presentation as to what these other examples are. The point I want to leave you with is that this is well beyond anomaly detection. This is looking at specific validated metrics that predict that the result of a clinical trial will be unreliable, that can be monitored by all of you in risk-based monitoring land. And we have some idea where these thresholds are and where we ought to be raising alarms and what to do to correct these problems in order to fulfill our goals of producing reliable clinical trial results. Sometimes these things are anomalous and sometimes they're not. So that brings me to a slide that I actually made while I was listening to others' presentations this morning, because I was struggling to understand in my own mind, why do I feel like an anomalous data point at this meeting? And I think it's because of this reason. I'm actually interested in what's on this slide, which is performance issues that impact the accuracy or reliability of the study results. That's really my goal. And detecting anomalies in critical variables is useful in detecting performance issues that impact the accuracy and reliability of study results, and that's good. However, there are a lot of data anomalies that have no impact on the accuracy and reliability of study results, and those should be ignored, in my opinion. And I realize that that may feel like provocative to some of you, and I'd love to entertain a conversation about that. An example of that would be propagated blood pressure measurements in an oncology trial, and I think I actually got this example from a very nice CluePoints paper, and I want to acknowledge the very important contributions that our colleagues from CluePoints have made to this literature. So I'm not endorsing propagated blood pressure measurements. That's scientific fraud and needs to be handled in a very severe way. All I'm saying is that that has no impact on the accuracy and reliability of study results in an oncology setting. And therefore, if you're focusing a lot of attention on that, you're probably wasting a lot of effort. Conversely, if that's the right word, there are often performance issues that can be measured and detected that are not anomalous. A good example of that is medication non-adherence. Medication non-adherence is normative in clinical trials. In chronic pain studies, the percentage of patients that are non-adherent to their medication is 45%. Those drugs don't work in patients that don't take them. And so here is an example of where just focusing on detecting anomalies misses an important source of loss of accuracy and reliability of study results. And instead, you need to validate a fixed threshold that you know predicts loss of assay sensitivity, if you want to use that term, or loss of accuracy and reliability of study results, and focus on detecting that and responding to that, whether it's anomalous or not. So I feel like that's why I feel a little bit anomalous at this meeting, and I hope I'm not getting too many people angry, because really this is the issue here: performance issues that impact accuracy and reliability of study results, and detecting anomalies is a means to an end and is an incomplete means to achieve that end. Of course, a lot of non-anomalous data is to be ignored as well, because that has no impact either. So my task that I've set for myself is, how do I detect performance issues that impact accuracy and reliability of study results? And then, of course, how to correct them, because simply detecting issues by itself is pointless as well, unless you do something to correct them. I know we would all agree on that. I'm going to skip that. So how do you actually do that? Or at least I'll try to share with you some thoughts about how we have been attempting to do that over the last few years. And the way that we do that, you'll all recognize this wheel, this sort of standard wheel of how we do risk-based quality management. We saw some very nice illustrations of that earlier. They weren't a wheel, but you get the idea. And the point I want to leave you with, I might actually not say too much more than that, is that each element on this risk-based quality management cycle can be linked through evidence as to whether it's related to the accuracy and reliability of clinical trial results or not. So for example, what variables do you want to monitor? And we saw some very thorough lists of variables that one can monitor through some of the nice earlier presentations. So you can look at all those, and all those, I agree, are important, but you can actually separate them into those that you can show based on evidence are relevant to the accuracy and reliability of your primary endpoint, and those that are not. And therefore, if you're interested in, as I am, in focusing your attention on accuracy and reliability of study results, then you can have some idea where to focus. Now, I don't want to pretend that we really know all the variables that impact the primary endpoint. This is a research area that's in its infancy. I'd be lying to you if I said we knew about all those, but what I will tell you is that we have a growing evidence library based on ongoing work of what variables do matter and what variables don't for the accuracy and reliability of your final study results. And you can go around the wheel and do the same thing all the way around. In interest of time, I think I'm just going to show you one case study of how this comes together, and then maybe have room at the end for a couple of questions. Maybe a point that I'll leave you with before I get to that case is that you can actually validate fixed thresholds on a number of variables that don't require statistics and don't require accumulation of data and don't require computation of p-values, but do tell you that there's a performance issue that requires immediate attention. So an example of that would be medication adherence. If a patient takes less than, and you can actually calculate based on completed clinical trials, how non-adherent can a patient be before it impacts your study results. And you can set that as a fixed threshold. And if a patient falls below that threshold in a week, you can trigger an alarm and do something about that, even if you only accumulated data on one patient from one site. And we have fixed thresholds for a number of variables that I won't bore you with today. Here's some data on that, but I won't take the time to show you that today in the interest of adhering to the timelines and not getting Eric angry. So here's just one little case about how it can all come together. This is a study that was sponsored by a sponsor whose name has already been mentioned during this meeting today and who have participants listening on the phone. This is a 5-site study in osteoarthritis of the knee conducted in the US, and this is data from 5 of those sites. One of the key endpoints was what we call staircase evoked pain, where the patients come to the research center and they stay there all day long, and every couple of hours they run up and down this stair according to the protocol, and their pain is supposed to get worse when they do that because they have arthritis of their knee, and we want it to get worse because that enables us to detect the efficacy of these analgesics in a more sensitive way than using the standard measures, which we have shown and which we have published in the literature. These yellowish bars represent what percentage of patients at these sites pain got worse after they did this staircase maneuver. And as you can see at this one site, site 14013, the patient's pain was not getting worse like it was in the other sites. So this was a problem. This measure was not doing what it was supposed to be doing. We had no idea why. So we were monitoring this because it was a key performance indicator, if you want to use that. If this wasn't working, then this endpoint was going to fail. So we were monitoring this specifically in this particular clinical trial. And this site was a statistically significant outlier compared to the other sites using statistical process control methods. We had no idea why, so we actually visited the site, sat there with clipboards, watched what they do, and it turned out that they were following the protocol. There was no protocol violation here, which is another good lesson. Not every performance issue is a protocol violation, and not every protocol violation is important for your primary endpoint. So be careful limiting yourself just to protocol violations. They were following the protocol, but in addition to that, they were having their patients walk around the research center all day long instead of sitting still, and that caused them not to, the patients were like impervious to the staircase maneuver. Their pain didn't get worse. Once we retrained that site to let the patient sit still between the staircase maneuvers, then all of a sudden this yellow bar started looking like what it did at all the other sites. Their data became normative, and what I don't have on this slide, but what I'll tell you is that before this retraining, there was no difference between drug and placebo at this site. And after training, when they were doing the procedure like they were supposed to, all of a sudden we saw a difference between drug and placebo that emerged that was similar to the difference between drug and placebo that was seen at all the other sites. So the moral of the story is that you can link risk-based monitoring to actual accuracy and reliability of the final results of your clinical trial if you monitor variables that matter, if you set performance thresholds that matter, if you retrain and normalize performance at that site, and you look at the ultimate reliability and accuracy of your study results to make sure that your maneuvers accomplished what they're expected to accomplish. And this is my last slide, and I think I'll just leave you with that final thought that you can actually do what the regulators are asking you and implement your measures in a way that supports the reliability of study results if you adopt a definition of reliability, if you harness your methods towards that goal, and make sure that you are optimizing your methods throughout your risk-based monitoring cycle to make sure that they're focused on the ultimate reliability of your primary endpoint of your clinical trial. The end.